So I called it Nonlinear Microscopy and Dancing Molecules. I wanted to give you a flavor of what nonlinear microscopy is all about and what we're trying to do with it. So the basic idea is, so microscopy, of course, is a familiar tool for biology, and it's used in lots of areas. So the question is, what's different about nonlinear microscopy? What can we learn? So we're essentially looking at new approaches to microscopy, and we're working with partners in pathology and radiology to look for various sorts of potential clinical applications as well as fundamental scientific applications for what we're doing. And so just to prime you, the idea of nonlinear optics is that you take light of one color, you convert it to a new color. And the reason that is interesting, or one reason that is interesting for microscopy, is it solves one of the basic problems of microscopy in that, so what we have here, there's this thing labeled linear focus, and what we have here is a cuvette with a dye, a laser is going through there, and for a typical microscope you have light that comes from all sorts of ranges through the depth of focus, so you have to play tricks to be able to pull out images with linear microscopy. On the left is basically what characterizes what happens with a nonlinear microscope. Essentially, what happens is the signal that we're looking at in terms of forming a contrast to form an image depends on the local intensity, and the intensity is strongest at the focus. You only get information from the focus of the light. We're looking specifically at third harmonic generation microscopy, and essentially what happens is if you take light, focus it with a microscope objective into a sample, you get no third harmonic signal. So in other words, if we take light that's 1,000 microns wavelength, which is in the near IR, the signal we'd be looking for is a third of that wavelength, so around 330 nanometers. And it turns out what you're sensitive are interfaces. So you look at things like membrane structures and various other features, and my pathologist friends tell me it's very similar to differential interference contrast microscopy in terms of the morphological figures you end up with. So this is not interesting. I threw this together quickly. But so here are some examples of some third harmonic microscopy images. So these are histologic sections. This is a histologic section showing a rabbit cornea. Some colleagues I work with were doing irradiation studies on rabbit corneas, and this is a histologic section afterwards. You can see a couple of features. I'm an engineer, I'm not a biologist, so I don't know the anatomy. I forget what this guy's called, but this is kind of a protein membrane. Probably someone here knows what this is. But you see this sort of protein membrane on the back end of the cornea. There's collagen in between. On the right, you see essentially the front surface of the cornea of the eye. These are just a couple of examples. Oops, fades away. This is an image of monolayer cultured cancer cell lines that were grown in a microfluidic chamber, and we're looking at trying to study morphological changes of these cells under various stress conditions. And so these are examples of third harmonic microscopy images that are relatively typical in terms of the sort of microscopy images you might be interested in. So the question is, what's the new angle that we're looking at here? And essentially the idea is we're looking for vibrational contrast. And vibrational structure essentially correlates with specific molecules. So the end goal here is quantifying and spatially mapping concentrations of molecules and molecular structures inside of tissues. And so this gives you a new tool into biological microscopy, or biology, at a cellular level and subcellular level. And the other thing that it buys you is potentially looking at particular signatures between, say, healthy and carcinogenic or malignant tissues, and various other ways to look for biomarkers. And the essential idea is shown here. Proteins and other biological molecules are very complex. This is a relatively simple molecule, SF6, but I put it up here to illustrate the basic idea that if you have this collection of atoms, there are certain internal degrees of motion inside of those atoms, and they have specific frequencies at which, in other words, in the time domain, they have a certain oscillation period that they go through motions in these internal degrees of freedom. And this is a well-known process, and Raman spectra are used in lots of areas. And the novel thing here is that by using nonlinear microscopy, first of all, you isolate information from inside of a cell. And secondly, by using the types of lasers that we use, you get much stronger signals, and so your sensitivity levels gets much better. And what we're working towards is levels that are relevant to protein expression inside of cells, hundreds and thousands of proteins. It depends on which protein and the condition of the cell. I don't know how to explain this data to this audience, but basically here's some proof that we can see some vibrations. It's relatively old data, probably not that interesting. If you want to know more about it, ask me. Like I said, I threw this together hastily. The other aspect that we're looking at is to be able to, in all of this work, we're using very short pulses, so pulses that are short compared to the vibrational timescales of this vibrational structure inside of these protein molecules. And it turns out that if you take these pulses and shape them in time in just the right way, you can drive specific vibrational motion and not others. And so you do optical pulse shaping. So for this work, we developed a new polarization-based optical pulse shaping that gives greater selectivity in terms of which vibrations that we drive. And so the fundamental problem here is there's a lot of interest and a lot of potential in terms of looking at vibrational structure at a subcellular level in this way. The problem is there's lots of vibrations, and so the question is, how do you pick out the molecules that you're interested in and look at them in a sensitive way? And one route to doing that is to shape the pulses in time to selectively only pick up or sort of pick up signals related to the molecules of interest. And some older data here shows that you can find pulse shapes. This is just proof of principle data for carbon dioxide that shows you can selectively excite relatively close vibrational frequencies by shaping ultrafast laser pulses. So to conclude, we're basically exercising this new microscope technology, and essentially we're kind of in a mode that we're looking for users, or looking for people that are interested in using this for basic science purposes, and looking for potential clinical type applications in terms of pathology or various other places of interest. And we're working with some of the people in the vet school here to develop some of these possible applications. And then the idea is that the work that we've developed here would serve as a prototype for potentially commercializing this work. That's all I have. Thanks. Any questions? Yes?
Can you describe some of the testing that you're doing with the vet school, like sort of specific applications they're looking at?
Yeah, a couple of things. I don't want to go into necessarily all the specifics, but one target that we're interested in primarily because they have small or relatively simple vibrational spectra are looking at, say things like transient free radicals like nitric oxide and things like that to be able to look at basic biology questions for some of these small molecules that are going around inside of inside of tissues. We're also looking basically trying to look at ways to assess, say, radiation exposure in sort of some of these clonal cell lines that some of the people that I work with have to see if we can look for ways to use signatures that we would pick up with this sort of microscopy to assess in a quantitative way radiation exposure damage to specific cells. And so the ultimate idea being, okay, maybe you can do something that's patient specific in terms of taking their cells, harvesting them from some tumor and seeing if you can tune the dose to the right way. But we're still at the stage of trying to look for a signature and various things related to those sorts of efforts.
Are the dynamics of this analysis such that you can see transient molecules like nitric oxide or superoxide? Is that possible?
Absolutely. So, I didn't tell you much about the lasers. The lasers are very short, 10 to the minus 14 seconds in duration, and come every 11 nanoseconds or so. So you can look at very fast transients. So any sort of transient that's nanoseconds, and most things of interest in, that you would look for that are transient species would be, probably shortest lifetimes would be milliseconds for most biological systems.